Hold on.
Yeah, go for it. So we should be warming up right about now.
Yep.
The
Yep.
thing's turning.
Okay. Cool.
Yeah.
Cool.
Wow, look at all that stuff he has in the background.
I know.
Okay, here we go. What the fuck's that? That's not us. Oh yeah, we're live.
Yeah.
And we're live.
What?
So Robot and the Bear rides again, and we are live streaming. We're actually live, and we're going to record, but we're also streaming live.
Yeah. Nice. Nice.
Yes.
We finally figured it out.
Yes. So we have special guest Gerald Pao from he's a scientist at the Salk Institute, right? So you are a PhD and an MD.
Yeah.
But you're at the Salk Institute. So first, before anything, tell us about what you do and what is the Salk Institute?
The Salk Institute is a biological research institute, and it was founded by this guy, Jonas Salk. And he's the guy who came up with the polio vaccine. And then he did that when I think he was in Pittsburgh. And then he didn't patent any of it. So he gave it for free. And then this organization called the March of Dimes decided, okay, at least we can give the guy an institute. And then the San Diego County gave the land, and the March of Dimes got the money, and they built this institute on the cliffs, on top of the cliffs in La Jolla, I think in 1960 or 1961.
It's a beautiful building from what I see.
Well, yeah, it's a pretty nice place. It's like you can I can send you a picture of it.
Cool. So what do you do there?
If you go on my Instagram, you see lots of pictures of the Salk Institute.
And Instagram that's why I know it, because I've seen your Instagram. It's Plurstr, Plurstr, correct?
Yeah, that's correct.
Yeah, at Plurstr. So can you tell us about what you do at the Salk Institute?
Well, I mostly work on other stuff nowadays. But
what other stuff?
Well, I'm working on downloading the brain into a computer and using doing neuroscience, computational neuroscience mostly, and we're taking basically movies of a brain of a fish, for basically a tiny fish larva that's about a couple millimeters long, but we can see every single brain cell, and then we're trying to download it into a computer so it can spit out the same responses as the real fish. So basically like downloading the brain. But then this came up, but I had
this meaning COVID-19, right?
Yes, COVID-19, yes. And then the virus that causes it is SARS-CoV-2. So the disease is COVID-19, and the virus that causes it is like SARS, coronavirus 2. So like the cousin of the original SARS. And then it's like I had a background of like 20 years or so of doing virology, and we also do like gene therapy. So I just put the old skills to use now, and it's like that's basically what we're trying to do now. So we dropped everything else, like I think mid-February, and then we started getting on this.
So how did you start it? Like your I guess to create a treatment or vaccine, or which one is it?
Well, actually, it's both. Right now, it's like one of the things that we're using can only. So we're trying to do two different things. One of them is just purely a vaccine. The other one is more like something that can do both things. The question is like we don't know how to use it properly yet. So it's like we're just trying to basically reprogram human T cells to instantly recognize and attack cells infected with the virus, so to clear them out. There's these so-called cytotoxic T lymphocytes, and we're using basically repurposing a technology that existed for cancer therapy for the purpose of antiviral therapy. But it's basically recognize anything foreign and then just get rid of it. The thing is, like, if you normally go through your immune system, like you have the flu, it takes you about 5 days to a week until your natural immune system comes up. If you were to have this, it would take you like less than a day to come up. So it buys you time until your natural immune system will take care of it. So you just jump started. But it's all about, it's a race, right? So if you jump started a race, you're gonna win, and you're gonna win faster. Kind of like that.
Hmm.
Wow. So wait, how did you guys get started, or how does it work where you're at? Are you allowed to just stop your other work and just jump right into something like this? And are you getting a government contract, or how does it get started?
Nope, we just used money that we had lying around. And then it's like, yeah, so we are trying to get money. There's one person from Silicon Valley who has pledged to help us out a little bit. Has given us like 50K. That's supposed to come tomorrow on Monday.
So that's weird. I mean, you would think it's not just 50K, that it should be like millions of dollars going to this kind of research.
Well, this will last us for like probably a month or so. But if we want to make it bigger and stuff like that, probably we need it. But I mean, we're starting with whatever we can, right?
Right.
Like we're like, but it's like there are whole bunch of like biotech companies, and our basically material providers are basically the people who synthesize little things for us, like synthesize the DNA for us. They have been like super helpful. They ask if you work on COVID-19, they put you in the front of the line. They make things that happen normally in the 2-3 week range. They give us to maybe like 5 days or less. They were able to put us right in front of the line, and they're very flexible who where to deliver and all these kind of things, because many times it's like, for example, your receiving department is out, so then maybe you have to have it go somewhere else. But that's kind of difficult because all these things work through purchase orders and have a lot of bureaucracy going behind, but they just cut right through it and just got it to us like right away. So it's kind of cool.
Awesome.
Yeah.
So the one thing I was wondering too is like, you've heard that you were hearing that this was all going on. How did you know that this was going to become something that you needed to work on as early as mid-February? Because that's still relatively early.
Well, I knew this was going to be a big problem. I think January 24th. So before that, it was very unclear what was going on. I think around, I think it was like around New Year's Eve, something came out saying that there are these unusual cases of pneumonia in Wuhan in China. And there was like associated with this, with this market. There's like a seafood market, although that's only seafood market in name. It has all kinds of other stuff.
Right.
But initially it was like, okay, there's some disease that's associated with that market. It's unusual, probably something new. We didn't know what it was. And then a couple of days at the beginning of January, I think like around the 5th or 9th or something like that, then they identify as a coronavirus. And I was like, okay, this is like something serious, because we had SARS in 2003, and that was serious. But they were saying that it's like, okay, it's only seems to be like transmission possibly from animals to humans, but doesn't seem to be human to human transmission. So you don't worry so much, because like, okay, you close the market down, the source of the animals is there. So then that's the end of it, right?
Right.
Yeah.
But then January 24th, there was a paper in Lancet. There's the Lancet and New England Journal of Medicine are the two top journals in medicine. And Lancet had a paper, January 24th, described a family who went to visit Wuhan. The family was from Shenzhen. Shenzhen is like next to Hong Kong, just across the Chinese border between Hong Kong and China, just on the other side of Shenzhen. And Shenzhen is like way bigger. It's like a super modern city.
Yeah.
It's like everything came up after 1985 or so.
It's a special economic zone, right?
Yeah, exactly.
Yeah.
So that place is like a super young, industrialized, and there's a family that went from Shenzhen to Wuhan, and they got sick. But it's like initially, I forgot which member, but it's like they went to visit Wuhan, and not everybody went to the market. I think there's like one family and an uncle or something like that went to the market, and then got everybody else sick. So the other people were in a hotel. They had several rooms in the hotel, and then traveled back, and then one after the other started getting sick. And then it's like, okay, that means that the uncle who was at the market got it, and then transmitted to the rest of the family. So now we have a problem. That's a big problem. Just as the first case that you see is a clear case of documented person-to-person transmission. And that was done by the Hong Kong University Hospital in Shenzhen. Some doctors reported that. So from that moment on, when that came out, January 24th, then we knew this was serious. And they knew it was serious too, because when they actually documented this, I think it was like the beginning of January, I think January 9th or something. By the time they, I think, got the last record on the paper, they talk about January 15th. So this thing was published from, finished writing on the 15th at the earliest, because the last piece of data was on the 15th until the 24th. So 9 days to publish in the top journal is crazy fast. This doesn't happen ever. Normally, if you have something published, it takes like a year or 2 years until from the time that you finish, submit the paper, the people review it, so on and so forth. That takes like easily a year. And this thing, in 9 days, it was out. So people knew this was important and had to get out fast. So when it came out, it was like only at that moment everybody realized, okay, this is something serious. The next question was like, are they going to be able to control it? And and I think it was January 23rd, the day before, is when China had already sealed off Wuhan.
Oh wow!
So it was like they actually moved relatively fast. They were really slow at the beginning, and it seems to be mostly because the local officials didn't want to have bad news to the central government.
Of course.
So they kind of suppressed it. But then apparently the central government was like majorly pissed that they were suppressing this, because the Chinese had like learned from 2003. They had an automatic reporting system throughout the country. And it's basically like anything that happens in every hospital gets automatically reported. They had a network of observation things that get reported. But the thing is, like, all the doctors were kind of, deferring to the local government. So then it's basically the stuff that's basically supposed to be a foolproof system of monitoring got broken by the local politics.
Mm-hmm.
So then it didn't get reported.
It's kind of happening that way here, don't you think? With the states.
Here is like, here was worse. Here was worse. And here was worse because we knew it and didn't do anything about it forever. So that's on us.
Yeah.
I mean, the CDC also has like, so I had like quite a few friends at the CDC, in the US CDC that's based, based in Atlanta. And there they have had, as since I was undergrad, at least that I know of, probably before, they had basically a permanent staff in southern China to monitor flu. So basically, every year you have like your seasonal flu, right? So you get a flu shot, but how do you know which strain of flu to include in the flu shot? Is figured out mostly by the people who are sitting in China, figuring out which are the flu strains that are going to come up. So the reason flu strain comes up is almost always starts in China.
Really?
Yeah.
Wow.
So the reason is because China has a lot of ducks and a lot of chickens.
We like our ducks. We like our ducks.
Exactly, yeah. So the largest population of so the new strains come from ducks and chickens, occasionally from pigs, but mostly from the ducks. So they actually have to see, okay, which is the dominant strain that's coming up now? And then they pick those and they tell the manufacturer to make it. So they guess like for the next season where it's going to be. So they have to sit in China and monitor the ducks.
So is influenza, or the flu, literally kind of rooted in birds?
Yeah, it's birds, pigs, but the largest source of the thing is in birds. So that's why they had like forever like a station. CDC had a station there, and then gives like some of the big companies like Sanofi, is basically Sanofi, and they and then they give this kind of thing to these companies, and then they pick out a handful of strains, and they pick those, manufacture those, and then get them out to.
Wow, that's how it works. That's so weird.
Yeah. So China, for example, doesn't benefit from a flu shot because it's too late for them.
Right. Wow. That's crazy.
So
flu shots are
Asia
based
in general,
China.
that's not going to benefit from a flu shot. But the Western world, like Europe, the US, we benefit from a flu shot.
Wow. Interesting.
That is interesting.
Let's fast forward. The reason why I was like, one of the reasons why I was like, oh, we got to talk to Gerald is because you have this picture of you holding a vial, and a vial looks like it's filled with sperm. but
yeah.
You wrote something about it might become something. I forgot the text on there.
Anyway, it's basically like it's a sort of like a prototype for a vaccine.
Yeah, you wrote maybe the first chimeric antigen receptor lentiviral vectors against SARS-CoV-2.
Anyway, yeah.
Which I only recognize words. I don't know what any of that means except for like maybe, the word maybe. I'm good with that one, but most everything else I'm
maybe
just like,
I don't know if anybody else is.
It looks important. It looks really important. So yeah, can you tell us more about that? Like,
well, it's basically like
it's what you mentioned earlier, right? It's trying
to make a vaccine, but we have like two different. So lentiviral vectors are basically they're based on HIV.
Mm-hmm.
So HIV is is the virus that causes AIDS.
Yep.
So I did my PhD in the lab that converted HIV into a tool that for gene therapy. So gene therapy is basically you convert the virus, gut out all the bad parts that cause the disease in it, and just make it something that can transport genes into cells. So that's basically like the HIV was re-engineered to be a tool for gene therapy. So for example, this has been very successful for some of the blood diseases like thalassemia, where you get red blood cells because they're missing a form of normal hemoglobin, and then you put a good version in and you get into the cells, and then you can cure the disease that way. Or you can actually have people who have like hemophilia and stuff like that, and then you can also cure diseases that way. So that is basically that is sort of like this is sort of like this is sort of the kind of thing that the technology allow you. But then a bunch of years ago, people started using so if you want to put a gene into a different organism and use a virus to carry it, this is basically what you use. And then this has been like a big industry also for cancer therapy. So Novartis has a treatment for leukemias and lymphomas, which is kind of a blood cancer, that is based on these lentiviral vectors. These lentiviral vectors carry instruction to your immune system to go attack certain things, and it tells you to attack the cancer cells. So we're actually trying to do like so we're actually making things that are kind of vaccines, just to produce the things to the immune system to attack, and that's basically what we're trying to.
So how did you know how to start? Like, it's kind of an abstract idea in a way, right? Or is this abstract to you?
Well, for me, it's not very abstract. It's like,
But you just had to use something that you kind of already had.
I wanted to make something that would be immediately practical. So then it's like I would use components that would be easily approved by FDA and stuff like that. So all these, drugs normally have to approve by the Food and Drug Administration. So they have like certain very stringent requirements of what can be approved and what cannot be approved, right? And normally it's test safety, and they have a regular clinical trial. But if you use components that are completely new, each component has to be tested. So basically, we just took the things like, okay, we want to put as little new as possible, and we're just going to use the components that already exist, and we re-engineer them. We are putting as few new components as possible to make something that can be like easily approvable. and still serves the function. So basically, it does this kind of thing. This is the kind of general approach that we.
So wait, so the therapy of using HIV, like the shell of an HIV virus to insert new kind of
so has been approved and is also used in multiple clinical trials. I think there are hundreds of clinical trials around the world doing that. So there are certain, like a lot of childhood diseases. There's also like that bubble boy disease that you have no immunity.
Oh wow!
So that
bubble boy disease.
John Travolta as the bubble boy. Yes.
So that now can be cured by that.
Does the bubble boy disease even still come up? Like, I haven't even heard about that.
Yeah, it's like called X-Kid. Yeah, and for example, there like the first case that there was cured is with a related other retrovirus. That was one done by a group in France.
Oh.
But they had a problem when the first versions of it, they did it not quite right. So, I mean, the kids would be dead otherwise. Right. They would not survive very long. So I think like 1 or 2 kids that they try to cure out of maybe, I forgot, something like 6 to 8 or something like that, got leukemia from the treatment itself.
Oh.
So, but 6 of them were okay, but 2 of them were not, something like that. And then they redesigned it and made it better with the HIV one, which made it better. And anyway, people have been like fine tuning it, and now it's working pretty well without the undesired leukemia.
Wow. So, your what is this called? Your vial of Gerald jizz. What is that thing? What is? Do you guys have a working title for it, or how does that work?
Well, this is something called lentiviral vector. Like the class of viruses that are used are called like lentivirus, like includes HIV. This one is based on HIV. And there's like, this is the type of vector. There was like a similar type of vector called retroviral vector. And this one is a lentiviral vector. So the advantage of the lentiviral vector over the retroviral vector is like retroviral vectors can only carry genes into cells that are dividing. And lentiviral vectors can carry things into cells that are not dividing if they're not growing. The majority of cells are not growing.
Okay.
When so that is like
you completely
that's
lost
basically
me.
like
I know. I'm like, okay.
Retroviral vector was
we love
kind of limited.
Science is sure cool, but we don't. I'm like, I don't understand most of science, so.
Well, it's basically like you can use it for very limited things. With the old version called the retroviral vector, you can only use it for very specific applications and very few cells, because these were basically very few cells in your body are dividing as an adult, or even as a child. Very few of those are growing. But but it's like a lentiviral vector can essentially go into any type of cell.
I see.
So then
I see kind of. But question: as you're working on this, how do you know that it works? Like did you?
We have to come up with tests to test this thing.
In your own building, right? Like you've already done tests.
Well, we're working on that. Yeah. Now we're like once. First, you make it, right? You have to.
Yeah.
As anything that you engineer, you have to make something.
But how do you know you're even on the right track? You have to test it on.
Yeah. So that's why.
Is there lab rats or something, or are you using rats?
There are different things. Like for example, like one thing that you can do is like, okay, you want to, as a vaccine, that what you do is like you inject this, and then after you inject it into the animal. And then after a few days, you check, do they make antibodies against it? And then you basically like say, okay, if I take some blood of the animal after a few days and then mix it with the virus, can the virus still go into cells? If the virus can no longer go into cells, that means that, okay, the immune response that you generated blocked the virus. So there's like what you call an effective. You have an effective. So we're working on that.
But you didn't test it on rats or anything yet, or?
We're planning to do that.
Okay. So do you get your own vial, or like do you get infected animals, or how do you get to that stage of testing it?
Oh no, we actually make and cell culture. So we grow cells.
Right. So okay. So you don't have your own. You don't have your own
DNA in. Huh?
You don't have your own vials of COVID that you can actually inject into a rat and then deal with and then test.
Well, we're using a COVID surrogate. Like we're using basically some genes of COVID and see that can attack those genes.
Oh, I see.
So yeah, because it's like in COVID, if you want to use the SARS-CoV-2, the virus, then you actually have to do this in biosafety level 3+. And then
what's that?
Also, yeah, actually you can. There are government sources that you can obtain it from.
Oh, I see.
So yeah, but there are people up the street that have it running, and that's that we will we will probably collaborate with them to work on this.
Wow. So what's your timetable on this to hopefully, if it, to get it through testing?
A couple of weeks, I think. Maybe we can actually say in a couple of weeks we can say, okay, this works, and then we probably have to figure out how we scale it up and how you want to produce it, how get approval, and all these kind of things. But first, like as soon as we can prove that this works, and then we can actually we probably have to find some kind of industrial partner who can produce it at scale.
But will you become a billionaire if it works?
No, because I already I'm not planning to, I'm not planning to make money of this. It's like,
it's the Salk Institute, right? You
okay.
follow the same
ethos. We're trying to see, like, we're just basically, try to, that this is the thing. It's like, so many of these therapies are super expensive. We want to try to make them cheap. We have to make them also like cheap enough that you can use it at scale. Like,
I mean, I was wondering if if it works, does somebody just give you a big bag of money?
That's up to somebody who give me a big amount of money.
It's kind of like
to improve this thing and like that. But it's like,
person out there goes, thank you. Here's a.
I mean, you were telling the story about Salk, who developed the polio vaccine. Does the Salk Institute kind of follow the same ethos of, like, kind of giving these therapies, to the public, or is it just a name?
Well, I don't know. It's like I don't deal at that level. I'm just the scientist. So these are decisions above my pay scale. Pay scale. This is like, I mean, I don't deal with any of this. I'm just the scientist in the trenches doing my own stuff.
So how do you know that there's not 10 other scientists doing this exact same thing right now?
Well, there are. There's probably hundreds of scientists doing this right now.
There are people trying different things, but it's like, I mean, I kind of try to do the things that were like not so obvious, and addressing some of the problems. And it's like, and this is not that what we're drawing is generally not considered a low-hanging fruit. So I mean, we thought through the immunology of the whole thing and what seems to be the problem of the virus and the type of thing. One of the problems that, for example, the conventional approaches we didn't want to try any of the conventional approaches because we know that people tried against other coronaviruses like SARS, and there was another one called MERS, which is Middle East Respiratory Syndrome, that people got from camels. That was also pretty lethal.
Wait, they ate camels?
No. They didn't eat camels. People had camels like in Saudi Arabia, and then is basically originally a bat virus that somehow got into camels, and the camels transmitted to humans. And since humans are in close contact with camels, we're getting it from camels, and it's called MERS. I mean, it's not only from camels, but that was basically like the distant cousin. So SARS is sort of like the cousin, and then the MERS is sort of like the distant cousin of SARS-CoV-2. So both of those were like serious diseases that are from coronavirus, but they were not super transmissible. And SARS was very manageable because people only started transmitting it when they became symptomatic. So you could just screen everybody for fever, and then you can just get everybody who was transmitting. The problem is like this guy is like, 2.5, maybe 3 days before you become sick, you're already transmitting. So then it's like, you cannot use the simple fever screening as a way to, quarantine people. So that's why it's sort of a perfect storm, this kind of thing.
What do you think about that Georgia governor who claims
I was just thinking of that.
Yeah, he claims just the other day he found out that it's communicable without having symptoms.
And it's ironic that the CDC is in Georgia.
He's not very well informed. The most amazing thing is like CDC is in Georgia.
Yeah.
Is Atlanta.
Yeah, I learned
That's
that from
kind of
World
like
War Z.
He's not getting good advice. What can I say? This is like,
Okay.
I mean, I'm sure he's busy being the governor and stuff, but you know, I mean, whoever's advising him is not advising him very well, I think, because you know, you should know these things, and it completely changes, you know, what you can do. And we know, for example, I mean, all this has been happening so fast. I've never seen any kind of epidemic where people investigated this thing like so fast. And you know, traditional publishing models cannot be followed because this thing is so important and is so wide reaching that people have been randomly posting stuff online. And and this is like the problem is like there's like a lot of crap out there. So you have to know what information is reliable and which information is not reliable. So that's been a challenge.
So that's where I step in because like I've been like into diving deep into like the weird shit of this of all of this on YouTube. And one of the questions, okay, so one of the things that I heard and I wanted to ask you about was this issue with blood type, like AB, and O.
So there's a report from China that is like the 0 or O blood type is less susceptible than AB. So AB is like worse, and like I don't know. This is a report, and it's like a single report. It's okay. It's basically just people just look. The numbers look worse with this. It was like, so I mean, for me, it's like I think it seems it's possible. Has not yet been reported by anybody else. Because normally in science you have like, okay, one person observes it, gets the information out there,
It's
and
peer
there's a
reviewed,
bunch of
right?
other people can confirm it over time.
Yeah, it's peer reviewed, kind of. Is that what it is?
I think
paper.
as I saw it, it was a preprint. I didn't see the review. So now there's places that are these called preprint servers, like bioarchive or chem archive or med archive, where people put their papers before they're accepted.
Oh, I see.
And they put it there. You know, a scientist in the field will be able to look at it and judge, okay, this is something good or something bad, or which parts are good, which parts are bad, because you have this training that allows you to judge this stuff. But you know, anybody, and they do a minimal review. Is basically okay, they make sure that you're not putting anything that's harmful out there, or conspiracy theory kind of thing, or that seems to have real data, and then you know, that is like looks like a real science as opposed to complete trash, right? So that's the minimal review they have. If it's basically like a conspiracy theory kind of thing, that is like, oh, this virus was made by some bioterrorism kind of thing, and kind of like make stuff up that is not backed by any data, people are not going to allow you to put it up there.
You're segueing great, because like the next thing I heard was that basically before Christmas in China, there were ads going up, you know, like looking for help at a bioresearch lab very close to the market in Wuhan that was apparently researching coronaviruses in bats. You know, and there's
a really famous lab there. There's a really famous lab called Wuhan Institute for Virology or something like that.
Right.
So they have a virology institute in Wuhan that was famous. Actually, they are the ones who first found the closest relative of this virus, and at least one of the authors was from there. That is like found in some cave in some bat in a cave in China, maybe like 2 years earlier, I think 2017 or so.
Well, apparently, like I think that the conspiracy theory is that the virus did not come from a wet market, but maybe from the institute, you know. And the Chinese government has been aware of this the entire time, and just kind of put the blame on the wet markets when they had to, and stuff like that. Because there was a patient zero that worked at that institute who has been kind of scrubbed from the books, blah blah blah, et cetera, et cetera.
Well, it's like, well, one thing that we know for sure. So there's a paper that analyzed this hypothesis that's been out there that people put that it was engineered or somebody put this virus together artificially.
Mm-hmm.
So there's a paper in Nature Medicine that actually analyzed this. Mm-hmm. And then look at how this thing was, how would anybody engineer something like this, given the knowledge that you had from SARS, mm-hmm. and given the knowledge that you had. And it's basically like everything that this virus has done to change itself is not consistent with the way people would engineer. So there's no real evidence that it's engineered, and it's much more similar to this thing. We can not say that is like it was. And also, it doesn't look like anything that escaped. It's not like directly from something that we know of. Mm-hmm. It's like different enough that we think that is natural. And there's actually it's been like 2 or 3 papers that conclude the same thing that have been through careful analysis show that this thing looks like something naturally evolved. It doesn't look like anything engineered. Because when you engineer stuff, you leave a little bit of you leave a signature behind. Like say when you, for example, if you were to build something, you have, for example, certain type of screws or certain things to put things together. When you genetically engineer stuff, you leave a little bit of stuff behind. And these and the signature of how you put it together is like it can be seen in there. And none of the stuff is there. So we really don't think that this is
I think the conspiracy I heard didn't say that it was engineered by the lab, but rather it was transmitted to a human through maybe some lab mess up, blood being spilled, urine being sprayed, whatever.
Well, if it's urine, then it's probably not, because we're seeing that the urine doesn't seem to have any virus.
Oh, really? Okay. Well, but basically the conspiracy is that the government knew about this the entire time and just didn't act on it, kind of like how our government is.
Well, I think it's it would be very stupid to shut down your own country like that. I'm sure economically it certainly doesn't make any sense.
Right. Well, I mean, I think the reason why they shut it, you have to shut it down once it goes out of control, right? But
so Canada has a biosafety level 4 lab that had basically a spill, but it's because of an accidental discharge, and then it's and they very quickly fixed the system so these things cannot escape so easily. I mean, after 9/11, the CDC also upgraded the security. Before, the CDC was not so secure, in the sense that it was secure if you went to the front door. But for example, if somebody came with a helicopter to the top floor and came down from the top floor, it was not very secure from that direction. So after 9/11, people started thinking about these things, and then secured the US CDC, because they had a virus that was super, super dangerous called smallpox. And smallpox, there's no immunity in humans anymore, because since the 1980s or so, nobody's been vaccinated against it. That's one virus that was exterminated from the face of the earth by humans. It exists in 2 places. One is in the CDC in Atlanta, and the other place is in a lab in Novosibirsk in Russia. So they keep those, and those 2 things, two samples, authorized samples in the world of that virus exist in these 2 places. And that is a way deadlier virus than this. That virus is 35% lethal to humans.
Wow.
And it actually and it transmits through the air way more efficiently than this one. So for example, the last case that was ever documented after it was exterminated in the wild was an accidental infection in a hospital. And it's basically somebody who's working on this virus in a hospital in London. And what happened, the virus got out because the containment was bad, and it killed It killed a journalist who was there interviewing people. It went up to the second floor to the air vent to the patients upstairs and killed some people upstairs. So there's a handful of people who died from that incident of this thing escaping.
What year was that?
1978.
Wow.
The year after it was eradicated from the wild. They actually had they brought a journalist and just wanted to show the journalist and ended up killing the journalist.
Oh my God.
Oh, jeez.
So is it true that this COVID-19 is it true that it's infecting men more, and if so, why?
So that's an interesting thing. So early on, people found that it was like it's infecting men more, but it seems so. There's I think it's like it was something like around 2-to-1 ratio of men to women, where especially they were like not infecting more, but it seems like they were like the serious cases where like the men were doing much worse. What turns out is like it seemed to be even more biased towards smokers than men. And it's because men smoke more. It seemed to be like, you pick it up to see it like there's more men are getting infected because more men smoke. And not only that, they found that the receptor, basically how the virus enters the cell, that molecule actually has much higher level. Once you start smoking, that ACE2, receptor for the virus, how the virus gets in, you have many more of those molecules on the surface of your lung cells. So it's probably related to that because now yeah, so now it's like people are thinking in other places where there doesn't seem to be such a large number of smokers, that does not seem to be, the bias seems to be much less.
Oh yeah.
So what about vaping? Has there been any studies about vaping and related? Because that's different than
smoking.
Yeah, I don't know.
Carcinogens.
There were some conspiracy theories of people thinking that vaping, the damage in the lung of the people, the vaping damage that people had like last year, where they was similar to this, but this is all BS. So yeah, people have not really looked carefully at vaping and ACE2 as far as I know.
Right.
Yeah, the way people do the experiments are really weird. Actually, you have to, you take a wad of cigarettes and then have a smoking machine and get the smoking to like monkeys or stuff like that. So you have the monkeys were literally smoking like a thing like this, like maybe like 20 cigarettes and like that.
Oh wow.
That's like really weird. The mouse ones I've seen is like they actually so you have a mouse cage that's completely closed, and then you take a whole bunch of cigarettes and basically a suction thing for that smokes automatically.
Just pumps the smoke into
pumps the smoke into the cage of the mice.
See, that's ridiculous. That is just fucking ridiculous.
That's how people get tested. Otherwise, you cannot deliver the smoke into the lungs of the mice. But it's like, I mean, the mice are essentially having like, secondhand smoke.
Right. Well, I mean, like, I think, I don't know. Like, I would actually love for there to be tests on, like vaping, just because, there are a lot of big question marks, over like, just how bad this is, I mean,
I think people are trying to figure it out. I mean, people only start paying attention to this like last year.
Right.
Yeah. Before it was like people just, didn't really look very much into it. And also like, I mean, after like tobacco stuff started ramping down, like when people smoking less, I think the amount of, as far as I've seen, like the amount of research that's gone into tobacco has gone down. And I think it's like there has been not much funding for research into vaping, but I think there will be some now as of last year.
So with related back to COVID-19, as a scientist, what's going to happen?
Well, this is a good question. So
yeah,
I think at this point is like what's probably going to happen, because given, so, like we have seasonal flu, right? So the reason we have like more flu, there's a flu season. And the reason there's a flu season is because like winter, when you actually have low humidity and lower temperatures, the transmission is better. And then people are hoping, there are a lot of people are hoping that, okay, maybe this virus is also the same. Other coronaviruses that are kind of cold type coronaviruses, they actually are also seasonal. So there's like a fraction of the cold viruses are actually other types of coronaviruses. They're very, very distant to the SARS-CoV-2, but they're also coronaviruses and they're seasonally driven. So people are hoping that, okay, maybe once it becomes warm, when we hit the summer, then this is just going to automatically disappear because the transmission is going to be bad. It's going to go extinct by itself, and that's probably going to happen. But what we're seeing in Australia seems, and New Zealand, which are having their summer now, and it's getting transmitted anyways, seems to suggest that, okay, maybe this will be here for good. It's like, and then eventually when you have 60 to 70% of the human population infected, and then the infected people, when they recover, they have like an immune memory. Basically, like, for example, if you have, many diseases that you have it once, you will have it only once, and
chicken pox type of thing.
Yeah, exactly, like chicken pox or measles. Once you have it once, you'll never have it again. And then you have immunological memory, and then it's like you can never be infected. So then it's going to become a disease of childhood. So basically, you're going to have, once enough children are born that have never seen this virus, this will be able to transmit in bouts related to the population, become big enough to be transmitted and maintain the transmission. So you're going to have epidemics periodically every few years or something like that.
But what about the cases of people getting the disease twice?
So that's not clear yet. So there are reports of that, and there are quite a few reports of that. So I don't know whether that's a problem in the testing. So the testing is not perfect. So let's say you got tested and then you show a bunch of times as negative, and then it comes back. But what is most worrying is a couple cases in Chinese healthcare workers, because they're monitored really well. And they also know more or less when they got sick and which events they got sick, because they kind of, it says, okay, this time I didn't put my mask on right, or like, this time I actually touched the patient. And so they can actually have a pretty good guess of when they got infected. So they can know they were tested a lot also. So it disappeared for a while and it came back like weeks after. So it's either they got infected again or they never got cleared out completely. So basically that could be like, you cannot distinguish those things very easily, whether basically you had like a low amount of virus that you couldn't detect and eventually came back. And we have a disease, a coronavirus disease in cats called feline coronavirus. And that one,
a good name.
Yeah. It's simple. It describes what it is.
Yeah.
Right.
So that disease is also really interesting in that is like, the feline coronavirus will, basically infect the cat for its entire life. It's in the gut of the cat and never gets cleared. So that's why you call it persistent infection. A persistent infection is an infection that lasts for the lifetime of the animal. And there are lots of viruses like that. So the human herpes simplex virus 1, which is basically HSV type 1, which causes you the cold sores, they just live in your nerves for your entire life. Right. Every once in a while, when your immune system is weak, you pop up and then you get a cold sore, and then it'll go away, and then it doesn't bother you. So viruses that evolve with their host for a long time, they tend to become very mild. So the long-term kind of thing is going to be, the virus is not going to cause very much disease.
You mean this
initially
COVID,
when a virus
COVID.
jumps species, it causes a really strong disease, typically, but then it becomes milder over time.
I've
Do you think there's going? Oh, go ahead, Luke.
I was just saying that, like, I've already heard that there's 2 different strains of the COVID-19 virus.
Oh, yeah. Can you explain that? I've heard that too.
So yeah, so there seems to be so genetically, You can so now people have sequenced over 3,000 genomes of this virus, like all over. And let me show you in a second here.
But do you think, back to your previous, I don't know if you can multitask easily. I was wondering if, okay, go ahead.
So, okay, you can see like they're like,
it's like Tetris, yeah.
It's like basically these are all the varieties of viruses that we know as of today.
Right.
And then you can see a branch here. This is like one type, and that's another type. So you can see they kind of branch in two different parts.
Right.
So that is like suggesting that maybe it's better to show it this way. It's easier to see. It's easier to see this way. You can see. You can see your left and your right. Right? So like there are two types that evolved.
Wow.
So yeah, we don't know whether there's like a big difference between the two. But it's like, yeah, we can observe there are two types, but it might be just because they are two types because they have a small change. And historically, it's like basically, okay, all the ones that went to Europe, for example, are of this type, and all the ones that went to North America are of this type. And then, but it doesn't significantly make a difference. Not a functional difference. Like they don't behave essentially different. Then it doesn't matter that they're different.
Then can you get one of each? Then can you get one of each? Or
yeah.
Basically,
yeah. I mean, like for example, like if you see, like the colors here that I show you, like for example, the red corresponds to North America, the green corresponds to Europe, the purple is like Asia. So you can see like where we actually have there's basically a very clear North American branch, but they're also very close to the Chinese ones. But the European ones are largely on this side, but it's not completely clean.
Wow.
So in the US, we actually have strains from all over the world.
America.
They have come from all over the world.
We're the melting pot. It's
we're
the melting
pot. Initially,
it's the curry from the world. It's the magical curry pot. We are that.
Yeah.
So you mentioned
we're the melting pot. It was like very clear. Like the early on, the Seattle one was like really interesting. The Seattle one was basically, it was the first one that you could actually see the virus evolving in real time in Seattle, the Seattle area, and then that spreading across. There were like other viruses coming into the United States directly from China. There were some that come from Europe, but they're from all over. And then, for example, the one in Seattle is the one that actually, I don't know exactly how, but somehow it made it to that Grand Princess cruise.
Oh.
So the Grand Princess cruise virus was clearly one that came from Seattle. So somehow early on, that thing came from Seattle and ended up in the Grand Princess cruise.
Wow. And
wow.
Although the guy they identified, the original person who actually died, is a person who lives in Sonoma County and went into that cruise. And then after he got off the cruise, he died. It was a woman, I think, she died.
Wow.
And then they were alerted after this person died, hey, maybe we should check. So the people who got sick afterwards from the next cruise. So there's a cruise that went to Mexico, and then the same ship went on a cruise to Hawaii. And then the people who went to Hawaii were the people who actually got alerted, and then it then became a shit show. Is
that the one that went to Japan?
No, the Japan.
That's a different ship.
The Hawaii-Japan connection is that's a different one.
Oh, there was a ship, right? That was in Japan. That was docked in Japan for a while.
No. That's a different one. That's
even a different ship.
Oh no, there are two famous cruises.
Yeah.
So there are two famous cruises.
They're infamous.
That's one was the Grand Princess, and the other one was the Diamond Princess.
Yeah, that's right.
So the Grand Princess is the SF-based one that finally docked in Oakland and got everybody off in Oakland. And then the one in Japan was the Diamond Princess that was docked forever. There was, I think, came from Hong Kong and ended up ended up in Yokohama, and it was there for a long time until
it made things,
came
and
back.
it got. I guess people got worse on the ship, right? Even if they were
Yeah, but we
sequestered.
learned a lot from that cruise because eventually everybody got, so the reason we know that they're asymptomatic people is because of that cruise. So basically, the Japanese ended up testing absolutely every single person on that ship. And the US CDC went there because I saw the paper in this journal called Morbidity Mortality Weekly Report.
I want that. Where do I get my copies? Jeez.
It's free online. You can actually.
Okay.
It's like the greatest
normally they're really,
title
really
ever.
boring, but in this case they were like, oh, now everybody's interested in it. But it's like, yeah. But they actually report on all.
I want a T-shirt. Do they have a T-shirt?
MMWR. It's like, it's kind of famous in the disease monitoring community.
Wow. Okay.
Yeah.
Anyway, so that's crazy.
So anyway, so in that journal they tested everybody, and then they figure out, okay, not everybody who we test shows any symptoms whatsoever. So that's how they know. And there was another city in a small town in Italy, a city called Vo, V-O.
Is that northern Italy that we
northern
keep hearing
Italy.
about? Gotcha.
Yeah, it's like a second. And then this guy from, I think it was Imperial College from the UK, from London, when he's actually Italian, but he went and tested absolutely everybody in the town. And that's how he figured out, oh, wow, there are all these people that show up that are basically not showing symptoms, but they have really, really high virus titers. So they have really, really large amounts of virus.
Wow.
But they were infecting their own families. So basically they show there's like everybody in their household was getting infected eventually. So there's like one person, and eventually everybody, the entire household becomes infected. So by this person who is not asymptomatic. So that was a problem. So I think those two are the key things that were found that make sure that people are okay. That's like the two lines of evidence from actual observation.
So do you think the Diamond Princess was that wasn't like a hidden experiment, was it, by any chance? I mean, they kept everybody
on. It's
not
a hidden experiment. It's like, I think it's a problem of jurisdiction and legal, and you saw Shin Godzilla, right?
Yeah, sure.
Yeah.
So I think it was a problem of Japanese bureaucracy.
Oh my God. But ultimately it actually helped science then, right? It actually helped science ultimately.
Yeah, in a way, yes, in a way. But it's like, here we're actually, the problem is also because, the ship, I think, was a British flagship, so Japan has no jurisdiction over it. So they couldn't actually make anybody do anything. Most of them are not Japanese citizens. So there are a whole bunch of different jurisdiction problems. And then Japan doesn't have a Centers for Disease Control.
Oh, they don't.
Oh.
No, they don't have. They have a Center for Infectious Diseases, but they don't have a American style or Chinese style Center for Disease Control, which is basically a government organization which is dedicated only to monitoring whatever disease may come up and stuff like that. So they're kind of embedded in all these different kind of ministries and different organizations, but they don't have basically a dedicated unit or a part of institute from within the government to do this.
Do you think they're going to have one now?
I think they will probably do this.
Thank you.
But Japan had like a lot of problems because of, everybody tries to be polite in Japan. Yeah. Sometimes it's like, for example, in Japan had a biosafety level 4 lab. That's the highest level of biosafety. But they never had any single biosafety level 4 organism in there because all the people around that area were complaining. So they built a lab that they never used for that purpose.
That's a Japanese NIMBY.
Not in my backyard. Yeah. Do you think because SARS never had and MERS, I guess, never had an actual vaccine, do you think there's going to be a vaccine for COVID-19?
I don't know. I don't have any. I don't know for a fact that it will be something that would be a good vaccine, because as you said, there has never been a vaccine for MERS or SARS.
Yeah, you actually told me that, so that's how I know.
Yeah,
you actually told me
17 years, right? 17 years that we had time to make it, but I don't know whether it's because of lack of effort, because people said, okay, SARS, it went away by itself, sort of. by isolation and stuff like that, we could get it out of the world. And then it's like we don't have to do anything about this anymore, or we can just. But this is a general problem of U.S. virology. U.S. is basically the biggest funder of biomedical sciences in the world right now, still, but not by much. I think China just overtook it this last year. But so the problem in the U.S. is like people says, okay, they're interested in a particular virus when it's causing the disease. But then it's like people lose interest in funding this thing maybe like 3 or 4 years later. So then it's like they wait until the next thing comes. So like in the 1990s, there was like 1980s, 1990s, 90% of American virology became HIV. HIV research, and everybody else who was working on other viruses was suffering because they were not getting money, they could not keep their jobs and stuff like that. So, there was very little other virology.
Right.
And then it's like, then SARS came up, and then people for a short time were interested in coronaviruses for about 3 to 4 years, and they lost interest. And then it was like pandemic influenza when the H1N1 came up. Then it's like people were interested in that, and then it's like lost interest again. And then there was like Zika, and there was first Ebola. People were interested in Ebola, and then Zika, and Zika kind of disappeared on its own. So okay, we don't care about this anymore. But it's like you cannot maintain. So if you want to have preparedness, you have to have a variety of things. You have to support a little bit of everything because you don't know what's going to come up.
Right.
So you should be ready for all the reasonable things that you can guess that you might want to be ready for. So that's a fundamental problem of. It's a little bit better in Europe, but this is the problem. And also virology became sort of like a less interesting thing. Once the people did the human genome, everybody wanted to do work on human genome kind of things and neuroscience and stuff like that. So virology became sort of like the.
Sorry. How hopeful are you in your own vial of magic?
I don't know. It's like I'm gonna give it a try, but it's like I think it's like if enough people around the world give it different tries and different people will try things, I'm sure. I think it's like a lot of it is that people, when SARS came around, people didn't care so much. There were not so many people who cared, and also vaccines are typically something that don't make money in the first place. So compared to drugs for cancer, especially, or for heart disease and things like that, medical procedures that are expensive. Or very common, like for diabetes, obesity, and things like that. Those make a lot of money. But for vaccines, don't make a whole lot of money.
Yeah, but this one right here is the jackpot, man. This one is it.
I mean, I think people realize how expensive this thing is. Like, I mean, just look at the stimulus, 2.2 trillion. You have to spend like that, right?
Yeah.
But that's not even taking account the damage, right?
If your therapy, what do you call it? Your own magic Gerald juice works. They're gonna make statues of you around the world.
Yeah, right.
Right? There's gonna be we're gonna make action figures. Luke will make a toy of you. It'll work.
Sell it in giant we can talk to Dub. We can talk to Dub about
it's gonna be amazing.
You'll make a pin of me and giant robot then.
We'll make a pin, but there's gonna be a Sofubi, man. We're gonna have a Sofubi doll made in Japan and China. It doesn't matter, and it's gonna be amazing. Yeah, but that's kind of amazing though. You're in a situation where you could actually cure the world with what you're making.
Oh, did you?
I can't do shit. Like, Luke, what can you do to save the world?
Nothing. Anything. I'm fucking useless. But
I'm useless too. You are actually a person that can do something, which is I'm honored to talk to you about that.
Yeah. Well, how do you feel about the comments from Dr. Peter Hotz, who was basically saying that they could have had a vaccine ready for this for coronaviruses, I guess. But because of the cutting, because of the things that you're saying, he was on Joe Rogan last week.
I didn't see it.
Yeah, but before that, he was at a Senate hearing, kind of talking about that, we could have been ready for this, but because of cutting to the funding to the research that he was associated.
I can tell you this. I can tell you a story that is very LA story. LA story is like this. So the world's expert in coronaviruses is this guy called Michael Lai.
Yeah.
Michael MC Lai, who was at the USC School of Medicine, like where the East LA interchange, the LA County Hospital.
Yeah.
Oh, yeah.
So that place, that place is like where most of the coronavirus molecular biology was worked out. How the coronavirus actually worked was actually worked out there. And this guy, Michael Lai, he got money. And then it's basically, he had this grant that lasted from 1990 to 2003. And that was like very well funded because his work was really good. But then they decided, well, coronaviruses actually are not a major, there's no medical need for it. It's like it doesn't cause any major human disease. So we're gonna stop funding him. So he lost its funding in 2003. He kind of moved over to hepatitis C virus initially because he was not able to fund his coronavirus research. And then he left in 2003 after he lost his funding. He went to Taiwan. But in 2003, later that year, then SARS came up.
Right.
Yeah. But it's basically people said, okay, there's not much medical need for this thing because we don't know any human disease yet. So that's basically like, that was the best coronavirus research in the world was going on there at USC.
Wow.
They just decided not to fund it.
So what do you do now? Like you go back to work maybe today or tomorrow, let's say. Are you still working on the same thing, or are you working on something different? Or
no. I'm still working on the same thing. These things actually take time. This is it's not a fast thing. You have to, like when you build, let's say, a car, you make one part, you make the engine one day, you still have to put the other things in, and you have to put the other thing. And then once you have it all made, you have to test it. And I say, oh my God, there's a defective part here. I have to switch this thing out. So yeah, so right now, like one of the things is completely made. The other part, we actually have to do that. Basically, we have to still switch a couple parts out to fix the problems there. And then we're doing testing to see how well it works. And once we know that it works, then we can actually say, okay, we have something, and then we can share it with people.
So are scientists from different laboratories and communicate with each other concerning this?
Yeah, there have been like some Slack groups and stuff like that. And there's like
what's a Slack group?
Slack is basically
it's software.
It's
right?
It's a piece of software. Yeah, it's basically it's like a chat group kind of thing.
Okay.
It's like a Facebook group, but for work.
Right.
Yeah.
The way the internet was originally intended to be. Right.
Yeah, sort of kind of like that. So anyway, so basically the couple of things, and it's like when you have a Google Doc and you share it with people and stuff like that.
Sure.
Yeah, so kind of like that.
So are you like chatting with people from Johnson & Johnson and people?
Yeah, depending mostly people that I know that are doing stuff, and then I talk to some people in Berkeley, some people across the street who actually have the virus, some people who are various people who are sort of in the space. But it's basically people that were my friends already before, and things. But because the actual number of people who are actually working on coronavirus before is like tiny.
Right.
Yeah.
Right.
And they were relegated to not so great places like Iowa and and things like that, because it's really hard to, like, for example, because it was not fashionable, it'd be very difficult for somebody to get basically a position to be a professor in Stanford or Berkeley, to be or UCSD working on coronaviruses, because it's okay, this coronavirus nobody cares about.
Right.
Oh, then it's like it was kind of not. It didn't look good.
Right.
People didn't care. So then it's like, now it becomes important, and everybody's going to jump on it, because it's also a good thing that everybody jumps on it, because everybody's going to try to solve this problem.
Right.
Because it became a major problem. But that's reactive rather than proactive.
Right.
So what do you think of this new thing in LA, at least Los Angeles, well, maybe the country, wear a mask, and then it's not a good mask. You're supposed to just wear a bandana, right? Is the bandana going to stop microparticles?
Well,
because that's what's kind of the worry, isn't it? The word microparticle came into the news like 2 days ago. It was never mentioned, right? Now it's microparticles, and I don't think. I mean, those things are tiny. So isn't that going to go right through fabric?
Yes and no. The thing is, like, if you eliminate, okay, this is the thing. Let's say your N95 mask. Your N95 mask is going to remove 95% of them. You know, still 5% chance of this thing going through, right?
What?
Yeah.
Really?
Yeah.
Yeah.
Oh, I didn't know that.
It's not
Is that what 95
5%.
meant? So 95 meant 95%?
Yeah.
Oh, I didn't know that.
Yeah.
Well, how come they don't just make a M100?
Well, to be absolutely sure, like because the way you make the pores and stuff like that, you make a material. You don't make uniform holes. You just make mesh, and it's like, okay. So then it's like, if I make a mesh of this average size, I'm gonna get rid of 95% of the particles of the particle.
I got a business idea. Let's go in on it. M97 masks. We got this. Me, you, and Luke. We got this business called M97. 97.
So there are filters that are 100% pretty much. They're filters.
What is that? That's just
like
that's just a bottle over your head, so no air gets in and you die.
No. It's actually almost there. You're almost got it. So
I'm a genius.
I'll show you, like, there is a BBC video of a South Korean ICU, and you're gonna see the nurses that work in there and the nurses and doctors that work in the Korean ICU. They wear basically a full closed, basically transparent respirator, but these are called powered respirators, and they have like these called HEPA filters. The HEPA filters will filter everything that, like, viral particle size. So for example, some of the equipment that we use HEPA filters that will filter anything that's the virus size. So basically, most of these viruses are something like less than 100 nanometers. So these actually can filter down to about 40, 30 nanometers, depending on which ones you get. But you can actually filter all of this stuff out. But it's because if you have to have this, like, really, really fine mesh, it's going to be really hard for you to breathe. So you have to generate a lot of pressure in order to be able to breathe through those things. So those are actually power respirators. So they actually run with lithium-ion batteries, which are pretty heavy, and they only work as long as the battery is powering. So they're actually basically they're enclosed. You have it, your head is inside it, and then it's basically I've used those things before. I mean, they're pretty, clunky to work with. But right now, like, my friends up the street who are actually working with the coronavirus, they're using that for the lab.
So, but what about a bandana? Like, that's what they're suggesting, or anything. They're
saying, if you actually have multiple layers, you're probably gonna get, like, my guess is, like, you're getting like 70% of it out, depending on how you put it. So it's actually helpful. It's definitely helpful. It's definitely helpful, but it's not going to be as good as an N95 mask.
So, but how late are we to the game?
Studies.
I mean, we're so late. This is just 2 days ago to wear a mask. Like, where was this, like, a month ago?
Okay.
Did they not believe?
So the Chinese had data on it. That they actually, it was mentioned already, and there's, like, I can send you an interview to the Chinese head, the head of the Chinese Center for Disease Control. He was saying it's a big mistake of Western countries not using masks. The masks actually remove a lot. And actually, there was a clinical trial about, I think about 3 years ago or so, that actually compared N95 masks and surgical masks for flu and healthcare workers. And they had like 2,900 or so subjects that they actually checked. And N95 masks and surgical masks were performing statistically indistinguishable from each other. They're about the same in efficiency. Because the majority of these things are like, like basically your spitball, but smaller, from your cough, from your sneeze, from just talking. This thing is just flying around. And there are all these videos now that you can actually see that.
Sure. I saw the NHK video, right? The NHK one. And it just shows two people talking, and that whole room is filled with microparticles.
Yeah,
that's crazy. I mean, just
micro droplets. Yeah,
it's micro, but the whole room is filled with it, and you know they're both just you're breathing it no matter what.
It's like you're making out.
Yeah. So tiny little bits.
So basically, Luke,
tiny little bits.
I have your saliva in me, Luke.
And
Gerald,
I have
you
yours.
too. And you too, Gerald. Probably
right now, I think that Zoom is more difficult, but you know.
Okay, yeah, that's true.
But everyone who's
do you think so? Do you think? I guess your a vaccine will be. I mean, it's a guess, but I mean, do you think a vaccine is going to be done on time versus us just accepting this thing and everyone gets it and we all live through it type of thing, or you know what I'm saying?
I honestly don't know.
You have no idea, huh?
I don't know. I can't say one way or the other.
But do you have a guess?
I think people are gonna try harder and try more sophisticated ways to try to make a vaccine. But I think it's like people have to fail a lot also, because I think the easy things that people have tried, people are retrying them without knowing because they don't know the history. This like the entire SARS and coronavirus stuff is like really obscure in the sense that not many people were paying attention to it. And a lot of people go, okay, this is the standard normal way that we make a vaccine. We're going to make it again like that and it should work. And you actually see all these like University of Pittsburgh, a bunch of other places, they say they have a vaccine. And a bunch of companies like Moderna and Novia says, we made a vaccine in 2 hours or 3 hours. But no, you just made something that allows you to try it, but it doesn't necessarily mean it's going to work. Just because you make something, it doesn't mean it's going to work. So until you know it's going to work, that's basically when people talk the fastest it can possibly be is like a year to 18 months to have it tried out, know that it works, and produce in sufficient amounts that you can start giving to people. Because not just showing that it can work in the lab, you have to give it to people in doses that are big enough that people's immune responses are going to be good enough. You also have to make it somewhere, right?
A lot of it, right? Billions pieces.
A lot of it.
So how long do you think realistic, like based on the information, because you obviously have a lot of information or you've ingested a lot of information about this virus, how long realistically do you think we're going to have to continue self-quarantining?
Well, the self-quarantining, I think, is like, it might be, I think it's like, if we did it right, like the Chinese did it, I think as like they have shown that you can do it in less than 2 months, right? I mean, it's clear from what you see there, and also from the South Koreans, that in 2 months you can bring it under control. After you bring it under control, then you have to do what's called contact tracing. It's not a very high-tech kind of thing, but it has a high-tech component, which is the testing part. So basically I said, okay, I'm going to follow this person and have this person, who did this person contact, who could be possibly like, and then you test every single person that basically came in contact with this person. And then if necessary, quarantine these people if you find something coming up. That's how we would normally deal with this kind of thing.
Right.
So probably I would think, for example, in California, you would have the California Department of Health or LA County Department of Health would have to generate these large teams that whenever something shows up, they just get on it and quarantine these people. And then you keep doing this enough, you might bring the entire thing under control. So basically, like, first you have to let it kind of stop and have like almost zero transmission, like what they have in China right now.
Right.
And then if you were to be able to do this like worldwide, you could actually eliminate the disease altogether.
Right.
If this thing that I told you before doesn't happen, there's a possibility that you have persistent infection. That's the person has the virus, and every once in a while it pops up. Then you have no control over it. Then you just it keeps coming back, and there's going to be no way for you to manage when the entire population it's like if you were try to chase everybody's cold sore around. Right? Everybody who has got a cold sore, now we have to quarantine everybody who is known. There's going to be too many of them. You're not going to be able to physically do it.
Right.
So if it becomes like that, and it's like that. The thing is, like, we don't know enough about this virus. I mean,
Right.
this virus has literally been in humans probably, now we're like April.
Yeah.
So
5
probably
months?
has been in humans, 6 months.
6 months.
6 months.
Yeah.
So you're
It's
saying
November.
like that the first, like the patient 0 could have came up in like October of last year.
November maybe.
Yeah.
November.
Probably
Okay.
November, most likely. October,
Wow.
November, yeah.
Wow.
Yeah, I mean, we can tell from how the genes mutate and how fast they mutate. We can kind of guesstimate when it happened.
Mm.
And there's basically, we guesstimated to be like October, November.
And do you think it came from someone like eating a bat?
No, I don't think it's somebody eating a bat. So we have like multiple.
You mean bat
viruses.
sushi?
Huh?
Bat sushi?
No, I don't think it's like bat sushi, but it's basically like probably somebody. So what people are really, really looking for is, you probably heard this thing about the pangolins.
Yeah, I heard the
Yeah.
pangolin.
Yeah.
Yeah.
So the pangolin thing, like, because people think that the pangolin might have been the intermediary host. So there's like the animal in between that transmitted. So I can tell you a story of another virus called Nipah virus. Nipah virus is like way more lethal than SARS-CoV-2. Nipah virus was killing, I think, like initially was killing everybody, and then became about, or I think roughly a third to half of the people who got infected. So this other virus is basically, how did this happen? It actually came from fruit bats, but the fruit bats never transmitted to the humans directly. And the first people noticed is these pig farmers were getting this. And then pig farmers were, getting this encephalitis, like the brain swelling, and they get brain swelling and fever and they die. So they were like, oh, this is really weird. So they thought something to do with the pigs. It turns out that it was like much more complicated. It's the bats, they're at the edge of the pig pens in the trees. They poop, it drops into the pen of the pig pig get infected, and the pig infects the human.
Wow.
But if you infect directly from the bat to human, it doesn't happen very readily. It doesn't work very well. But if it goes through the pig, it infects the human very readily.
Wow.
So the pig added something that basically we actually know what it is. It's like they basically, it made the virus, it coated the virus with a bunch of sugars. When it went through the pig.
Sweet.
And that's what allowed it. Yeah, it sweetened the deal.
Wow. This is like, that's science humor. You should do stand-up science humor. Just scientist humor.
Wow. So you think maybe like a couple more, like I think Bill Gates came out and was saying 10 weeks or something, like 10 more weeks.
Yeah, I mean, people are actually, it really depends how well people stay at home.
They're not doing a good job.
No, they're not.
Not they I guess.
Actually,
I'm doing all right.
Numbers, LA was the worst in California.
For what?
San Diego is better. Bay Area is better. LA is not so good. Actually, LA is basically the liability.
The numbers? By the numbers, you mean? Just how many
in terms
cases?
of how well the you're flattening the curve.
Oh, yeah. No, I think, is it stuff like, is it Florida and New Orleans are really rushing up fast now? Yeah, they're going to overtake California.
Doing terrible.
So.
Actually,
they're doing really bad.
Right,
Georgia will be next.
So there's a system that exists that the hospital thermometers send their readings to basically to a website, and then they look at anomalies, basically unusual numbers of fever, and then it's basically they saw unusual number of fevers in Florida after spring break.
Wow.
Nice.
Yeah, of course.
But didn't see that coming.
There's so in California, it's an unusually low number of fevers now. But that's probably because you killed off the flu also. So because people are doing, because people are staying away from each other, they're also not passing the flu around. And also people are trying to avoid hospitals, obviously, right now. So then you don't get readings. But if you get bad enough in Florida, people are seeing
you go to hospital
in Florida. That is like people are getting.
So when the numbers are in, it means you're really fucked. like you're in a bad situation, because that means you're at the
Probably
hospital
right now.
Oh,
Given the situation, that's what I would guess.
because if you're
actually
sitting
sleep
here,
over
if you're
so well-
sweating with a 100-degree fever, you're at home, so there's no readings for you.
Yeah. Or if you go to the
Wow.
hospital and get your reading and it looks, oh, I know you have a lot of them showing up in the ER. I think most of the people who are gonna show up at the ER now are gonna be for something fairly serious.
Yeah. So
I
in
don't
the
think
future,
people are gonna catch.
what's the way to prevent this? Is it just don't eat duck? Stop eating duck, everybody.
Stop eating penguin.
chicken.
I don't think eating duck is the problem. The problem is like you have to have a good monitoring system.
I mean,
of course,
kill
those
every
markets
bat. Let's just kill all the bats everywhere. No more bats.
That's not okay. That you might actually have a different virus coming up if you do that. So you remember there's like this dengue fever.
Yes, that comes from what, a bug that a bat would eat or something.
Yeah, and the bugs would be eaten by the bats that keep the dengue numbers down. So you have the mosquito.
God damn it!
Yeah,
so,
you need the bats.
I mean, getting rid of bats is probably not going to be basically there's two major things. One is human encroachment. Humans are expanding their agricultural area. So basically, the one in the Nipah virus one, what I told you about the pigs, that's basically people going into the jungle and making the pig pen like right next to the jungle. So then it's like the tree is next to it, and then it can drop it. But if
no trees next to the farms, cut down every tree.
No, it's basically like also
okay,
it's
not
also
work.
not good because like
it's not going to work either.
If you make the animals escape from the small patches of forest, then you also can. Like so, for example, there's a bunch of viruses that happened in Brazil. They were in Brazil. One of
more viruses.
Pentagonal virus. Yeah, there's actually escaped vaccines that evolve in the jungle, and as people encroach into the jungle, people got infected by those.
Well, at least it's not an Asian virus. It's a Brazilian.
Yeah, actually, like people can actually trace that back to a vaccine.
Yes, at least it goes okay. And then the one happy thing I have out of this conversation is, well, not happy, but I mean, this Asian virus thing isn't new, right? The flu shot, you said it comes from basically China, right? It's whatever's in China.
Yeah,
it's been an Asian virus all along.
There's tons of Asian viruses, and they're African viruses. Like for example,
but the flu shot is Asian.
Yeah, man, middle country, baby, middle country.
We've been here. We infiltrated the world,
essentially. Yeah.
Wow. Okay, well, that's more bad
it's
news.
Always been, there's been agriculture.
Luke, should we just beat ourselves up at this point?
I don't think. I think you're fine, Eric. It's I think the Chinese people should stand up and just take the blame for everything.
You know what? There should be videos of Chinese people slapping themselves in the face, and then everybody just put it up. There'll be 1 billion videos of Chinese people doing that, and there'll be no more violence against Asian people.
No.
Redone it, right?
No. Actually, it's like
people didn't used to care.
Yeah, but it's big news now.
Yeah, I know it's big news now, but people become people used to not care because the viruses used to be called before after the place where they were found first.
Mm-hmm.
So, for example, Ebola is the Ebola River. But the cousin of Ebola is Marburg, is in Germany, because
this
the
is actually
Institute of Virology where it showed up. Huh?
This is actually the Wuhan virus, right? So
yeah,
Donald Trump was right.
In the old days, people will probably call it the Wuhan coronavirus.
So Donald Trump was right.
No.
Oh, it's like people moved away from this standard
because he was calling it the China virus. He wasn't calling it the Wuhan virus.
He did for a while, and then he probably forgot what Wuhan meant. When he was like China. Yeah. Anyway,
but there was there's a funnier virus. Well, not funnier virus.
There's that science humor again.
So there's a science humor story of around 1993. There was a big drought because there was a La Nina year. And
what's that?
The opposite of El Nino. El Nino years when we get a lot of rain in Southern California.
Oh, okay.
So there's the opposite called La Nina years where you have drought.
Mm-hmm.
So when in that year, like there was a drought, I think it was 1993, and then there was a virus that showed up in an Navajo Indian reservation.
Whoa.
And then it killed, initially it was killing 100% of people. And then it came from these North American deer mice.
And this is funny, huh? Okay.
Well, no, listen what happened to the name. So anyway, so the virologists, they found it in this place called Muerto Canyon. This Muerto Canyon. So they want to call it Muerto Canyon virus. But then the Navajo, basically the Navajo didn't like that. So they said, okay, we're gonna call it the Four Corner virus, because this is the four corner, like New Mexico, the four corner. So then people complained, there's like, you should not call Four Corner virus. So then it's like the virologist got frustrated. It's like, we're gonna call it Sin Nombre virus. And you know what Sin Nombre means in Spanish? No name.
No, but it's a movie. What's it? No name?
They call it, so officially that virus called Sin Nombre virus.
It means no name.
No name.
It means no name. No name virus, because they got frustrated because they couldn't use any name.
Wow.
So they're fine, we're gonna call it this.
And they did it in Spanish to make it sound like it's from Mexico.
It's
completely a sham. Yeah, that's so us.
Do you see this other thing on BBC that is like the Mexicans want to close the border, so the U.S. North like Americans do not go.
So they're going to pay for the wall now.
Yeah. They're making the Americans pay for the wall.
Oh, okay. The other way around.
Well, they already have been. So
yeah.
Wow, this has been great. Thanks, Gerald. Freaking yeah.
But how bad is this really? Like, how much worse could this all be? Could it even be worse?
Yeah, it could be worse. I mean,
this is bad, but could it be way worse? Should we be thankful of anything, or are we just like?
I think we're not gonna reach the. Okay, if we thought that it was gonna be as bad as Italy or France or Spain, they're getting about around 10% death rates.
Right.
So they're getting 10% lethality because they're basically their healthcare system is overwhelmed. I read the things in the Spanish news that are like,
this guy knows Spanish, by the way.
You know Spanish? Wow.
Yeah. I was born in Madrid.
Oh, okay.
Yeah.
Nice.
Yeah, this guy's he's look at him. He's from Spain. Anyway, so yeah, you're thinking it's not going to be as bad as North Italy or whatever.
Well, if we don't do the social distancing right and manage things right, we can get to that point. It's like, I mean, if you get those rates, that'll be like something like 330 million people in the United States. If you have like, if you don't do things right, you can end up with 33 million people dead. And it's like, for example, my friends in Madrid are telling me every single person personally knows somebody who has died.
Wow.
Basically, virtually every person personally knows somebody else who has died from this.
Right. Wow.
So if you get to that point, it's gonna be like, if 1 of every 10 people is dying from this.
Yeah.
And also, like, it seems to be the initial thing that we were thinking that only the old people are being affected. Yes, most of the old people are being affected, but now it's like, that's come to the US. Now you're hearing, little children getting it and dying. There was like some teenager in LA that died, right? And there's like I see reports everywhere. I mean, they're not such huge number, but that doesn't mean that nobody's immune.
Are we going to really do you believe in that 100,000 number for the United States? They said 100,000 people are going to die.
I think that's very possible. I think that's kind of an optimistic
really
number. Yeah.
Where are we at now? Isn't it like it's under 10,000, right?
No, we're we're much higher than that.
In terms of deaths? I have no idea.
Yeah. I mean, like, we've already, I don't know. I mean, let me look.
And it just seems like Los Angeles is
I don't know exactly the numbers that we.
It just seems like Los Angeles's numbers are not as high as I thought it would be at this point.
Yeah,
I thought we'd be higher.
I mean, you have to thank Gavin Newsom,
right,
for getting the statewide, shelter-in-place order early.
It was late anyway, in my opinion, but okay.
Yeah, I mean, Yes, it can always be earlier.
Okay, it was early for white. It was early for white man, but not for Asian man.
Yeah, that's I think that's a fair assessment.
Because I was like, why is this happening? Why are we out? Why is a marathon happening in Los Angeles? Like, why are they running a marathon on March 8th? You know? And I was thinking, that should not be happening.
I mean,
right?
it's like I think it's like, to me it was very clear, like the end of January, that this was going to happen sooner or later, and we should get our act together.
Yeah,
because of like that was clear that like it seems like the Germans also prepared early on. I see some reports that the Germans were trying to ramp up testing, get everything ready, get their hospitals, fill their hospitals with ventilators and PPE, but they still were not. They couldn't get it in time. But it's still better to be prepared than not, right?
Yeah.
I mean, it's like.
So, are we going to be okay? That's the question.
Yeah, we'll be fine.
I think if we behave, we're gonna be okay.
I honestly think we'll be fine.
As a society. I mean, like,
this is coming from
San Diego. I feel that people are doing all the right things. Like, for example,
the numbers are low.
The homeless people in the convention center with good separation.
Right, where Comic-Con's going to happen, and we're all going to then go there and die in July. Yeah.
Well, when it comes down to Comic-Con, you're going to be there.
I know, man. I'm going to get whatever residual something.
Well,
they're going to go. They're going to say, "Oh, it's going to be. It lives for 2 months on certain surfaces. The wooden tables at Comic-Con. Oh, that one. That one's grippy, man. That virus is going to live there forever."
I honestly don't think Comic-Con's going to happen.
I don't think so either. Do you think Comic-Con will happen? Which is mid-July, I believe, or third week of July, maybe second week of July.
I don't know.
Probably not.
Like, everybody's hoping this thing will be done by June, but
No.
Yeah.
No fucking way.
It's hard. That's like way too many people in one place, and that's kind of wrong thing to do, probably.
Yeah.
Or if this thing is still around, and I think it's probably gonna be. Yeah, I don't know.
Everybody who's into Comic-Con, Gerald Pao, scientist at Salk Center, says sorry.
No, wah.
That's my best guess.
I honestly think this entire year is just a flush. Like it's fucked. Like until like some kind of vaccine comes around, like,
or the Gerald's therapy, whatever you want to call it. You got to have a name for this thing.
Vaccine.
Make a cool name for it, man.
Yeah.
It's such a bunk name you have.
I don't have a very creative name for it, but when
really
I know it works, I'll ask
Luke
you about
Luke is good at this. Luke would be good at naming.
I am not good at names. My characters don't even have names, so like I
figure you title
what's
art.
The name of the bear. I still don't know the name of the bear.
Doesn't have a name. It's me. It's me. It's basically me. I'm trying to find this. There's this great video that I like put on when I work that like just has all the statistics of like how many people like have been infected, how many people died, how many people.
Go to the John Hopkins website.
John Hopkins.
That's the one that's.
John Hopkins has a website that.
It's no longer that little 17-year-old kid who made a website. Remember that? It was such a big deal. Everybody kept promoting it. The 17-year-old kid,
like that?
He made one, and then now it's like nobody cares about that anymore. It's John Hopkins.
Is it a YouTube stream? Because I was looking at this YouTube stream.
No, it's just a John Hopkins website that has statistics worldwide. But
there's a whole bunch of them. There's a UW one. There's a
see, this is the one that I've been looking at. It's this weird kind of like stream, and it has all these numbers and maps and everything like that. Yeah. Jeez. I mean, we've had like 69,000 deaths, but that's worldwide.
Yeah.
So
I don't know how many's here, but it's over 10,000? That's already too many.
I'm waiting for them to get back to America, and I'll let you know.
Wow.
And it has really somber, quiet music playing on in the background, so they can just work.
Oh,
wow.
It's like a more stylish version, so it allows you to work.
Yes. Well, I mean, like, I'm really into the study beats, and so like which I play when I'm working, and then I'll now I've been switching it up with this guy, which is
So do you go back to work today, Gerald?
Yeah, we're probably gonna go in the evening.
How many hours are you working on this? Is it like desperation, or are you just kind of like going at the?
Well, we try to do as much as we can, but certain things require time. For example, when you make stuff, you have to grow up bacteria. They have to grow up to make the DNA that you have to use, and then you cannot accelerate that. That takes like 18 hours to grow, so you have to wait for it. You just put in the afternoon, you come the next day and pick it up from there. But there are things you put into cells. It takes like 3 or 4 days until this thing. So, but we're trying also to minimize the, we're trying to be good citizens and minimize contact and maintain social distancing at work. So we're actually trying to go there as little as possible, but still get all the stuff done.
Because I keep thinking of, is it David Banner, the Hulk? Like everything he did was like you do something. No? Or is it Bruce Banner, David Banner? Bruce Banner. Yeah. But every time he did something, he could look in the microscope and that thing's moving, and he's like, "Oh shit, it's happening!" Right? But for you guys, for the reality is, you do something, you're like, "Fuck, be back in 18 hours, see what happened."
Well,
okay. So not much desperation, because that guy he was sweating like desperate, trying to do stuff.
Well, here's the question
anyway,
I have.
Guess not.
How do you get the DNA? Is it DNA or RNA that you put inside the shell of a HIV virus?
So we actually put DNA into cells, and the stuff that goes into the viral vector, that's actually RNA. So we actually use the cell to make the RNA that goes actually into this, and then the tube that you saw is basically the viral vector, the actual virus that contains the RNA inside.
So how do you get the RNA inside the virus?
Oh, so it has something called a packaging signal. So the viral components, there are a bunch of proteins that make the shell, and the shell has a component that recognizes this little sequence, has a specific sequence, and then we make the DNA that contains that specific sequence, and when it gets made, it automatically goes in there.
Wow.
So that's basically like that's how the virus is like. So we just use the features that it already had.
So how do you neuter a bunch of HIV?
We remove most of those genes.
And it's not like some weird nanotech thing, is it? Or
it is kind of like, this technology was first made in its very original form in 1996, and it kept improving until around 2002, 2003, and then by 10 it was pretty mature. And since then it has had, like, you can add more things to it a little bit, but it has not changed that much since then. So for about 8 years, people just kept making big improvements in safety and design and stuff like that. And after that has been like, you add some bells and whistles for particular use, or there has not been that much. Then it's basically like using the stuff, but it's like when people in computers, people came up with x86. Now it's like, okay, we're just gonna make it faster and stuff like that, or make it cleaner, or make it something like that. But the essential design is the same. So yeah.
Question before we run or let you go or whatever it is. Can you tell me what did Junko do on this? Did she work on this too? Junko?
Yeah, Junko.
Ogawa? So Junko does art at Giant
majority
Robot
of the
2
work.
sometimes. Oh my god. So Junko does art, and we have her in art shows sometimes at GR2. And I know that she's worked on this too, but I had no idea what her role was. I thought you were doing it all because I'm a dude and I expect dudes to do everything.
Actually, but she's done.
I thought she just hold the vial or something. No, she does. She does work. Okay.
Course, yeah.
Go Junko.
Yeah, go Junko.
Awesome. Give props to Junko because
I will. No, she's done the majority of the work.
Awesome. And she knows what she's doing.
She knows what she's doing.
God, she plays it so like
coy.
She plays it like she knows nothing. It's amazing.
She knows everything.
I believe her now. I believe Junko. I'm always like, yeah, you don't know anything. Nope. I'm like, I guess what? Did you clean the gloves? What did you do? She's doing the work. Wow.
Yeah.
I'm impressed.
Yeah.
Junko Ogawa.
Junko is
awesome.
Junko is like a top grade molecular biologist.
Really?
Really?
Yeah. Oh, but she's all. But okay, so that's awesome, and it's rad that she has time to do art. That's crazy that she actually gets into. She's into art.
She probably doesn't have much time now.
Between, or like when we have to wait for stuff. Yeah.
She's just drawing.
Yeah.
Wow.
Wow.
Amazing. I'm even more honored.
Mm.
Yeah.
Yeah, she
That's
actually has
awesome.
she drew a picture now on COVID-19.
Oh, really?
Really?
Yeah, one of her
Cool.
pieces is a COVID-19.
Oh, yeah. I saw it. It's a beast.
Yeah.
It's in the show.
Yeah.
Yeah, I saw that one. It's amazing. Yeah. Cool.
Oh, she also had the other one, the Japanese thing. What was it called? What do you call that thing that you put in the front of your store?
Oh, Amabie,
Yeah, Amabi.
right?
Yeah.
Amabie.
Yeah.
Yeah. I drew one too. So,
You drew one too?
wow, I just put it on my stories. That's it.
I'm gonna make a Amabi pin.
No. I could. I don't know. No one would care. But so
It's popular.
any last things, Luke? Any last words, Luke?
I think like most of the questions that I had were pretty much covered. So
yeah,
I
awesome.
appreciate
we'll get more
time.
if you have time. We'll get you again if we get more and more confused because
before you become like like
oh
the star of the COVID world.
One last thing I have, Gerald and Junko, save us. That's all I have to say. Save us, save the world.
Yes.
Save us, please.
We'll try. Somebody
This
will
sucks.
probably
Save us. No, fuck them. You guys do it.
Right.
We'll
Actually,
try.
you
know what? Asians saving Asians. Let's do it. It came from Asia. It ends with Asia.
Yeah.
Asian American. I don't care.
Yeah, Asian American.
Any Asian.
There you go.
Yeah, any Asian American. Yeah, starts in Asia, ends with Asian American.
Asian American.
I'll take it.
Nice.
Like the way it immigrated.
Yes.
Yeah.
Yes, exactly. Take care of it.
Wow. Well, actually, that's kind of like like I just watched a video, and where they were kind of trying to explain like how COVID, or 19 works.
You're not off the hook.
And then like how like apparently like they think Johnson & Johnson is trying to block the virus by blocking the hooks, with like, is that like
that's one of the therapy attempts and things like that.
Mm-hmm.
And there's so the receptor of the virus is called angiotensin converting enzyme 2. Mm-hmm. Is actually a target of blood pressure medicine. So this blood pressure medicine that blocks it, people think that you could actually repurpose the blood pressure medicine, certain types of them, to block the virus from entering the cells. But it's going to be hard because there's so many of them, and it only needs a very, very short time to go in. So it's going to be hard to flood that system.
Right.
So yeah, they're trying, but many people are trying. I can send you a list of the things that people are trying. There's like a ton of things.
Another one was like, I think they were trying to take a docile virus and give it like a hook, and I don't know. It was a weird video.
This sounds like Katamari.
Yeah, kind of
pushing a ball around.
Pushing that ball.
No, I mean the majority of like the so-called live attenuated virus is basically a mild version of the same virus. That's actually probably the most effective vaccines that we've ever had. If you can make a version of the real virus that has a bunch of mutations that doesn't allow it to grow very well in humans, but yet it looks very similar, then the immune response against it should be really good. So that is basically probably your best bet in terms of getting a vaccine. I think that would probably be the best possible vaccine if somebody can make one of those.
Make it,
Gerald.
Start today. Today is the day. Start today. Save us and save the world.
Yeah.
End it, please.
We'll try.
Please end
We'll
it.
try.
Thank you. That's better than I can do. Only thing I could do is sit in here and watch.
Yep. All I could do is sit back.
You can inspire people with GR2.
Okay, I'll try. But thanks, Gerald.
Dude, thank you, Gerald.
Thank you so
You're
much,
welcome.
man.
Dude, thank you for your time. I appreciate it.
Talking to you guys.
Thank you.
No,
thank
pleasure
you.
was mine.
All right. Thank you. Robot and the Bear out.
Robot and the Bear out. Are we still streaming?